The few or only known viral disease which is completely eradicated. The strain is no longer available except for two countries, one in US, which is preserved in a very high security laboratory, and one is Russia. So as we progressed, we all might have heard about the polio vaccine, and we now know that all the kids who are infants and babies get the polio vaccination. The present polio vaccine is the oral polio vaccine, but in 1930s, scientist named Jonas Salk, he developed the first killed virus vaccine by killing it using a formalin. So that was the first modern day vaccines which had a mandate to be tested in humans before getting it into the market. And it revolutionized the field of virology and immunology. In fact, the Salk Institute established in US is after his name. And that was the first time when the live attenuated vaccines were being introduced. And then apart from Salk, Sabin also did in 1960s. What he did was he found that the heat-killed or the formalin-killed viruses are having some adverse drug reactions in people, and that was causing deaths. So he thought of attenuating the virus, that is by weakening it by growing in a non-specific host for several times. So when we do that, the capacity of the virus to replicate actively gets reduced. And with these two guys' efforts, the global polio vaccination effort was kick-started, and it has gone through until 2021. India has been declared polio-free. Only now 3 countries in the entire world, which are Afghanistan, Pakistan, and Nigeria, have polio. Rest of all the countries are free of polio. So that's a second feather in the human virology and vaccinology after smallpox. So from 60s to 80s, the smallpox eradication program has been continued. Many countries declared them smallpox free by 80s, and India declared it by 85. And in post 1980, we had the future of vaccination programs. So until now, I have given the historical perspective. I will now dwell into the scientific advancements and a little bit of immunology as we go ahead. So what actually happens when a person is inoculated or when he is infected? So basically, our immune system gets activated. There are 2 arms of our immune system, which is innate and adaptive. So innate immune system is what we are born with it. And these are cells. These are first line of defense. It could be our sweat, our saliva, our skin, and certain cells which engulf the virus. And when they engulf the virus, they train the adaptive arm of the immunity to identify that if a second infection happens, then you have to create a system in terms of antibodies to combat these diseases. So as you see in the chart here, and this concept is so good and so well developed, now we understand it, that we use the same concept to have vaccination. And that's how you might have heard about the first vaccination, the booster dose, and how the memory response stays. To give you some examples, like smallpox vaccine, which we all have taken, at least my generation has, people born after me have not. We all carry the antibodies for smallpox for a very long time. Not all the antibodies are for lifelong. And that's why you may question that why we keep on getting new coronaviruses and why our body gets come back. Because none of the antibodies which are circulating in our body is lifelong. But in order to boost our immune system, we can give a booster dose. That is, after a gap of certain time, we can give back a certain inoculation with a specified dose to reactivate the immune system. So for this part, the memory cells, and there are B memory cells and T memory cells. B cells are those cells which produce the antibodies. So antibodies are these Y-shaped structures which has a specific binding link for a specific pathogen. So they are so accurate that now we have monoclonal antibody-based companies which are making the antibodies specifically targeting for particular types of cancer. Apart from B cells, there are also T cells which activate the immune system in such a way that they provide the long-term immunity. So this concoction of the activation of B cells and T cells together, they combat us, they actually provide us strength to fight any disease. And this is happening even as we speak. Imagine somebody among you is having cold, which is a family of influenza virus. So the antibodies are being generated even now, and your cell body is getting adopted. That's why we see, and we have a common saying, as we grow, it's okay for children to get sick because they get immunized, they get natural immunity. So this concept of immunity is very important to understand how to make a viral vaccines. Certain vaccines only need antibodies, but certain vaccines, they just need to have a very strong effector T cell response. So moving on, just to make complex things very simple, these are some of the examples how the body's immunity is activated. So there is passive immunity, as the term passive says, it goes slow and it's not too activated. So there is natural maternal antibody, which comes from our mother's milk. There is immunoglobulin in that, and there are antitoxins in that. In the active immunity, where we have infections, specifically seeking the natural viral infections. So here we have attenuated organisms. So what they do is, not all the strains of viruses are so strong, but when we get an infection, we already have a prime and boost response. We understand the memory. And when a second infection happens from the same species, which is much stronger, the previously existing antibodies help us. Now, here is a caveat to it. We all know a very important disease, being in India, the tropical disease of dengue. In the past 2 decades, dengue has even progressed not only in the tropical parts, but also in the cities because of the way we are living, because of stagnant water, and mosquitoes are the carriers. So there is a concept here that even though we know that antibodies help us to fight, but in dengue, infection from dengue, one gives one particular type of antibody, but the same antibody will not help us in the dengue 2 infection. So this is something very new we understood in the past 15 years, and that's why now people are trying to make a vaccine specific for dengue, which covers all 4 dengue serotypes. At present, we have 4 dengue serotypes prevailing in the humans. So the antibody-dependent enhancement makes the dengue infection even more difficult. So if you go to a doctor, he would always question that, did you have a previous dengue-like symptoms? And is this a second symptom? And the reason being, because the second dengue infection can be very severe, and the existing antibodies will help the second infection to grow even better. And that's how our platelets go down. So this information and this knowledge is very important to understand what kind of vaccines we need to make. For example, the cloned microbial agents, like for the hepatitis, it's a surface antigen which is important for providing the antibodies, immunization. So whatever hepatitis B vaccine we took, it is recombinant DNA technology produced hepatitis surface antigen, which is inoculated in our body. And there are other passive immunization happening from the same family of viruses, like common cold. So there are multivalent complexes which are growing. So coming to one of the most important pandemics, this is the influenza pandemic. It is called the mother of all pandemics. We have witnessed one pandemic of corona, but the deaths of 1918 Spanish flu is nowhere comparable to what coronavirus has. It is estimated that around 50 million people in entire world died. And this is a historically interesting fact that when the World War I finished, it is at the same time that the Spanish flu emerged. It is also important to share an anecdote. It is a misnomer. Spanish flu did not start in Spain. It was World War I, and most of the countries had strict guidelines for their media not to report anything. So there was a lot of censorship. But the US had won the war, and the armed forces were walking back from their camps, from the ships, and it was at Cap Kansas, which is in USA. In 1918, you see a black and white picture with all the soldiers lying in a big aerodrome. This was actually an airport. They were brought back from the ship and from planes, and they all got sick at one time. So at that time, people did not have an idea. Scientists did not know what exactly is causing these deaths. It looked like starting with the common cold, but severity stepped in and everybody started falling sick, and at least 40% mortality rate was there. And on your left, you see some scientists holding these eggs. So it is that time when, and the cell culture was not so much developed there. There were no sophisticated labs. But some of the smart scientists understood that an embryonated egg is equivalent to one single cell, and it is rich in content, and the influenza virus grows very well in these eggs. And that's how the first egg-based vaccine concept came here. It is still prevalent and it is still done in many countries. That's a different concept. I will not deal further how to make the vaccines in the egg, but I will tell you later when we come to the exact vaccination process. Why influenza is very important, and it's one of my favorite viruses, is influenza has a big family. It has 4 cousins. So influenza A, which infects basically only animals, including birds. It doesn't infect B. But it's influenza B, which mainly infects humans, and it's influenza C, which infects humans, pigs, and but not more than A and B. So now we have identified, and the nomenclature, if you see a repeat of H and N, these are the surface proteins of the influenza virus, H being hemagglutinin, N being neuraminidase. So the number H5 or N1 in the first, it includes the hemagglutinin 5 and neuraminidase 1. This is how the nomenclature has evolved. So the influenza can be highly pathogenic or low pathogenic. So these subtypes of viruses are still evolving. And because of this, the concept of how viruses are evolving was very well understood. So influenza became a model organism, which later helped to understand even the coronavirus evolution. So coming back to the pandemic, we have witnessed at least 3 major pandemics with influenza alone in the past century. The first one was 1918 Spanish flu, which was caused by H1N1. It was from bird to humans. And you may ask, how do you know that it was from bird? Because the sequence of these strains are still available. And with the emergence of sequencing technology, we know certain context, certain strain structures which are present, and we can map them to the birds. And this was the first indication where the zoonotic or the transmission from animals to human sources was happening. And it was so severe that 50 to 100 million fatalities happened, and these are documented. And while I was doing my master's, I did understand that they have some archives. And since this was National Institute of Virology, they found an archive where they found that at least 2 people in the entire city of Surat, in one family, in each and every family, died because of this. So this was during the British era. So one of the most affected cities were Surat and Mumbai, because these were ports where the ships were coming, and the virus came from US to India, along with the humans. After that, in 1957, we have witnessed H2 and N2 influenza virus, and these 2 viruses completely resorted. So they were 2 different strains. They got a chance to mix. We still don't know whether they mixed in human or they mixed in any animals. And this information is very important for us, because even the conspiracy theories or the sequencing data says how in the present coronaviruses evolved from the markets in Wuhan, and how the virus from one particular species could jump and be a part of the second species. So the concept of reassortment was known, and then in 1968, again, there was one more pandemic caused by H3N2, and this pandemic caused 1 million fatalities. It's much less than what happened in 1918, because by this time, there was a mandate to produce annual influenza vaccines. So, but the question remains, are we anyway close to the next pandemic? The answer is yes and no. Yes, because now the travel is happening, people are interacting more, there's so much of globalization. So the viruses travel along with the humans. And no, because we are much better prepared with our technology, with our sequencing data, which we share with the countries. And that's how organizations